Today we're going over the pieces of the microscope. So to get started, on your microscope, here, we're going to have a base at the bottom, an arm of the microscope. You'll have the stage right here. You'll have your condenser, which is right below the stage itself. We can see it right here. So what this does, we can move it left or right and you can see the dial turning. If we focus on the stage itself, as I dilate my condenser, more light passes through, whereas when I close it, less light goes through. See that by looking right here on the microscope. So this is where light is going to come up and hit your sample. So again, all the way closed, all the way open. To adjust the height of your stage, you have two adjustments. You have the innermost ring, your large knob, which is a coarse adjustment. So it'll move the stage up or down, depending on which way you turn. And then the fine adjustment, which is on the outside, does the same job as the coarse adjustment. As you can see, the coarse adjustment is still turning, depending on which way I turn the fine adjustment. However, this again is for fine adjustment in the microscope for focusing. Next, we have your objectives. So you have your objectives on the microscope, a 4x, a 10x, a 40x, and then you also have a 100x oil immersion objective. So this objective lens is only to be used while you're using oil immersion. All the other objectives, as you can see when we zoom in, all the other objectives, such as the 40x, so the only objectives that you're going to be using with oil will say on the objective tube itself, oil. Otherwise, your 40x, your 10x, and your 4x are going to be just when using your sample and focusing in. So, now that we've covered our objective, let's talk about where you should start. So every time that you put a sample onto the stage, you're going to want to get the stage set such that you see some of where your stained sample is, and you'll always start with the lowest magnification lens. So in this case, it's our 4x lens. This is our scanning lens, our scanning objective. So since most microscopes currently made are typically parafocal, once I have my sample resolved using my scanning lens, I can then increase the magnification to the next objective, and it should be in relative focus. Now with this, you will still need to use, again, your fine adjustment knob right here to make any adjustments. So once we have our sample fully in focus, then we also have the time to apply the oil. When you're applying oil, what you need to do is first your sample's in focus on 40x, then you move a half stop between your 40x objective and your 100x objective, and you apply oil. To apply the oil, simply get your oil dropper, and only a small amount of oil is needed. So as you can see, that's a little too much, so I'm going to let that come off. And now what I'm going to do is get a small amount of oil and dab it on. So I'm going to turn down the light so we can see the oil on the slide itself. So you see, as you can see, there's only a small droplet of oil. And then we'd get our oil objective lens, and we'd swing it into position. Now mind you, this isn't in focus at the moment. So now what I'm going to do is I'm going to rotate my oil objective into my droplet of oil. And as you can see right here, the oil and the objective are actually touching. That's what we want. So that limits the amount of the refractive index between the objective and the sample. So this way we can get better resolution. So now that we've covered the objectives and when not to use oil, remember, oil is only used with the oil objective. Now that we have that covered, there's another objective or another lens on our microscope that we need to be concerned with, and that is our ocular objective. So you have two eyepieces. You have these guys up here, and these are your ocular objectives. So on your ocular objective, they'll typically have a place where they say what their magnification is. So when looking at this, we need to consider we have two objectives on our microscope. We have our ocular, our eyepiece objective, and then we also have the objective that's in the microscope itself. So the reason I say in the microscope is you can always change out your ocular objectives. So with this, you have an ocular or eyepiece that has a certain magnification. For purposes of this video, our objective is 10x for our ocular lens. For your objective lenses here, again, they vary. So to get the total magnification power of your microscope, what you will do is you'll multiply your objective that you have on. So right now we have a 100x objective oil immersion lens, and you multiply that by 10x, which is the resolving power or the resolving magnification of your ocular lens. So 10 times 100 is 1000x. So that means here you'll have a total magnification of 1000 times, or 1000x. So remember this when doing labs or writing up what magnification you used. You always consider what your total magnification is, or you could write down separately the magnification used for your objective lens and your ocular lens. Okay. So one last thing that we need to make sure to talk about is proper maintenance of your microscope. So when you are done, for example, with oil immersion, what you need to do is you need to clean your lens, your objective. So what you use is your lens paper and your lens cleaning solution. So you take out a sheet of lens paper and you apply a little bit of lens cleaner. Make sure you give yourself enough room. Move this to the side. And now, just lightly rub against the objective. So you can use this lens paper and lens cleaner with the other objectives as well, with your eyepieces. If something, some kind of dust, debris, common things that might get on your eyepiece, for example, makeup. So you can use your lens paper and your lens cleaner to remove any of these residues. So you want to make sure it is lens paper because things such as filter paper are actually too abrasive, and what that's going to do is it's going to scratch up the polished surface of your objective lens. So be sure not to use anything other than lens paper when cleaning your microscope, and also your lens cleaner. So today what we're going to be looking at are two different types of mounts for applying sample onto a slide. So the first that we'll cover is a wet mount. So standard wet mount, get your sample and what you'll do is you'll take a drop, small drop, and put it in the middle of your slide. Now that I have applied my sample, I'll take a cover slip and what I'm going to do is lay this just across and then let it fall. So when you do this, what you'll be able to see is your wet mount or that droplet will actually spread across the slide from the weight of the slide compressing the droplet. So now that you've successfully mounted your drop, now you can visualize it on the microscope. So you'll then take your sample and starting at the 4x magnification, you'll then find, you're going to resolve and then once you find something, then you're going to increase magnification, and then for these, the highest magnification that we use is our 40x. So the reason why we wouldn't use oil immersion is we have a cover slip. So the whole purpose of oil immersion is to lessen the amount of refractive index between your sample and your objective, since we have a cover slip between our sample and our objective. Using oil immersion would be kind of useless. So for that reason, we wouldn't use oil immersion when we're having a cover slip. So now that we've covered our standard wet mount, what we're going to do is do a hanging drop mount. So we first have a different kind of slide. So this is a specialized slide called the depressed slide. So I have this inverted right now so we can kind of see the depression or that circle in the middle of the slide itself. So what I'm going to start with is I'm going to take some Vaseline and apply around the edges of this drop slip. In your lab manual or other methods of doing this, you could also use gum. However, Vaseline will suffice for our needs. And as you can see, we want to make sure that the Vaseline covers this square area. This will be really important when we apply our hanging drop. So now, as the name kind of implies, this is a hanging drop mount. So what that means is we take our slide, and what we'll do is apply some of our euglena that we've picked up from the bottom of our sample container. And what we'll do is we'll take a drop. So you then take this droplet and invert it and carefully place it onto your slide. And as you can see, as in the wet mount slide, we had our solution spread out across the slide. In this hanging drop, it is basically localized to where you have that drop, that droplet that you applied onto your cover slip. And now this will allow us to visualize our euglena.